Great. Okay, so I'm going to talk to you now. This is going to be spread across 2 lectures. So I'm going to talk a little bit before lunch and a little bit after lunch about polygenic indexes. Several people have asked what's the difference between polygenic indexes and polygenic scores and polygenic risk scores. Those are all the same thing. And so I'm going to describe a little bit at the end why we use polygenic indexes in these lectures. It's sort of a new term, like new enough to me that I'm likely to say polygenic score several times through this lecture. I'm going to try to maintain the same terminology as I speak, but if I slip up and do that, understand that, again, these correspond to the exact same thing. Okay, great. So before we dive into talking about polygenic indexes, we're going to talk a little bit about SNP heritability. Raymond used the term SNP heritability several times in his lecture. We maybe should have had this discussion before Raymond's lecture. But now we're going to talk about what SNP heritability is first. So remember our additive model. So we have our phenotype, and then it's described by a linear combination of SNPs, and the additive factor here is meant to be a linear combination of SNPs that maximize the predictive power. What's the most that a linear combination of SNPs could predict in our phenotype Y. And so this is different than the general genetic factor, which is how is SNPs altogether, not just linear combinations of SNP, describe Y. And so this gives rise to a notion of what we're going to call narrow heritability. And narrow heritability is how much of the phenotype is described by a linear combination of SNPs. Right? And so the heritability you might get from some other estimation model might be bigger than this because we're constraining ourselves just to this linear combination. So when we say narrow heritability, this is going to be slightly smaller than the total heritability of the phenotype. So the reason, okay, so then there's a further problem. So I've assumed here that this sum corresponds to every single genetic variant in the genome. There's J of them. However, when we have genetic data, we don't genotype every single SNP. There's maybe rare variants or things that just aren't the SNPs on the SNP chips that we use or things that we can't impute well. And so in genome-wide data that we work with in practice, we can only measure some subset of all of the SNPs in the genome. And so the number of SNPs that I'm going to say are the ones that we can see. I'm going to refer to as cap k, which is less than J. And so I'm going to index these SNPs just from 1 to cap k. Okay. So then imagine that instead of, for the additive genetic factor, we said imagine how much all genetic variants in the whole genome could explain the phenotype, right? But now I'm saying let's just consider the SNPs that we have genotyped. How much can that subset of SNPs explain the phenotype? And so if we did this, then there's going to be different weights associated with each of the SNPs just because some of the information is going to get passed around to the SNPs we can see from the SNPs that we can't see. And so I'm going to refer to this A sub SNP as the additive SNP factor. And so the additive genetic factor is how the portion of the phenotype that is explained by all SNPs. The additive SNP factor is this term here, the part of the phenotype that's explained by the genotyped SNPs. And so then we can re-express this model where the phenotype is now the additive SNP factor plus a different amount of error, right? So now there's going to be some residual in the phenotype that we can't capture just by the set of SNPs that we have. And then this is also going to give rise to a notion that we're going to call SNP heritability, which is what I was saying we were going to talk about before. So narrow heritability is the fraction of the phenotype that's explained by all SNPs. SNP heritability is the fraction of the phenotype that's explained just by the genotyped SNPs. And I have this picture here, which is similar to the one that Dan showed about GWAS talking about kind of what the relationships might be between, or how there can be differences between SNP heritability and the additive genetic factor. And so in this picture here, I say imagine that we have 5 SNPs in our genome. So we have a 5-SNP genome, very, very simple organism. And the height of these bars is the effect of each of these SNPs in the additive genetic factor. And so, this SNP is associated with an increase of 5 units, if we're controlling for all SNPs at once. However, maybe in our genotype data, we don't observe SNP3 for some reason. Well, if SNP3 is correlated with SNPs, 1, 2, 4, and 5, then in our additive SNP factor, some of this signal from SNP3 is going to get passed around to the SNPs that we do observe. So even though we don't observe this SNP, we can observe, the effect of this SNP through the SNPs that are in LD that we do observe, And perhaps there's some fraction of SNP3 that just, isn't passed through, and so there will be some loss in the heritability of the additive SNP factor relative to the additive genetic factor. And so, I tried to represent that as this hashed section here. So that's the portion of SNP3 that we just don't capture with our 4 SNPs. And so, as a result, what this means is that the SNP heritability will generally be smaller than the narrow heritability. Yeah?
So that SNP3 will be, we can also observe it in, not observe, but it will also be somewhere in the narrow heritability also, when we are doing all SNPs.
Yeah, so the question is about, so would SNP3 be in the narrow heritability? And the answer is yes.
Because that's all SNPs.
Because the narrow heritability captures every SNP, even if we don't see it. Yeah, so, and those are the things that, maybe twin studies are trying to estimate. So, we try to infer what the SNPs are in a kinship model. But if we have genetic data, then, we can only think about the effect of things that we see. Okay. We happy? I think that's, okay. So SNP heritability, there's ways that we can estimate it. There are a bunch of methods that have been developed to estimate it if you have individual level genetic data. There are also approaches that have been developed to estimate it using summary statistics like LD score regression. So LD score regression estimates SNP heritability and not narrow sense heritability. And so that's one powerful, fast method that's been developed. So I have a bunch of claims here. SNP heritability is an upper bound on the total predictive power of the SNPs identified. It's an upper bound of the total predictive power of SNPs identified by GWAS. Okay, yeah. So the point here, sorry, I was realizing that didn't say what I thought it said. The point is, we do a GWAS, we find 30 SNPs, that are associated. And we said, how much of the phenotype is explained by just the 30 SNPs that are genome-wide significant in our GWAS? And I'm saying that that's going to be less than the SNP heritability. Why is it going to be less than the SNP heritability? So let's think about EA1. EA1, we found 3 genome-wide significant SNPs. And I said, how much of the variation in education is explained by those 3 SNPs? A big amount? Yeah.
So I'm assuming it's an upper bound because there are so many other SNPs that are in LD that these few SNPs would capture. So in a sense that we don't know those SNPs are, but our estimate actually is an upper bound in a sense that it captures the effects of other SNPs in LD with those.
Yeah, well, so that's a reason why those SNPs maybe do better than, so it's not just, those 3 genome-wide significant hits are just going to capture they're going to capture the effect of that SNP and any SNP in LD. But why is it that there's going to be still a gap between that and the SNP heritability? Well, what is SNP heritability again? Yeah, well, all genotype variants, right?
Yeah.
And so is that going to be more or less than the heritability explained by just 3 variants? Yeah, it'll be a lot less. And so that's the sense in which it's an upper bound, right? There are a lot of SNPs that we have not yet identified in GWAS that probably actually contribute to heritability, but we just haven't found them yet. And so there's a gap there. And as we find more and more SNPs, maybe we'll get to the point that we're capturing more and more of the SNP heritability by the SNPs that were found. And for height, it's actually been really interesting to see, they're capturing more and more of the SNP heritability. And, the gap's getting quite small between how much of the variance in height's explained by genome-wide significant SNPs versus how many is explained by all SNPs that we observe. Okay, so that's the first point. The second is that SNP heritability is a lower bound on the narrow heritability. So why is that? All SNPs, yeah. And I think the principle here that I just also want to highlight is that narrow heritability is a function of the SNPs that we're using. So if we throw more SNPs into, if we genotype more SNPs, then SNP heritability will increase. If we remove SNPs, SNP heritability will decrease. And this is also going to have implications for polygenic indexes when we talk about that in a minute. I guess this is the last little point. In the limit, if we had sequencing data, so we genotype every SNP in the genome, then the SNP heritability should converge to narrow sense heritability. And the idea here is if every SNP is genotyped, then SNP heritability is the how much of the variance is explained by every SNP. And there's a working paper online now that claims that by using whole genome data, they're able to capture there is no longer any difference between SNP heritability and narrow sense heritability. So it seems like for height we're getting, if not there already, we're pretty close. Great. So we know what SNP heritability is. How do we estimate it? Well, there's two groups of methods. I've already kind of talked about this. There are methods that are based on molecular level data, and so that includes things like Gremel, LDAC. There's a method by Alex called RDR that also estimates SNP heritability. Each of these give a slightly different flavor of SNP heritability, but depending on your definition of what your SNPs are and what your effects are, they'll give you different answers. There's also methods that are based on summary statistics. So LD score regression will give you an estimate of SNP heritability, and all you need are GWAS summary statistics. SumHer is also a method that I think Raymond mentioned in his talk too. So there's another. These are just examples that people see, and each of these rely on some sets of shared assumptions, some distinct assumptions, and so sometimes you'll see differences in the estimates across these methods, and that's probably just due to the distinct assumptions across the methods that you use. Great. I also want to make another point about what if there's confounds? So in the model, well, I'm not going to go all the way back, but in the model that I showed before, I kind of just ignored these confounds. But really, we need to think about what we've included as controls, right? And so let's say that we did a GWAS, and we didn't include parental genotypes as controls. And let's say we didn't even use PCs as controls. Well, what's going to happen is that the genetic effects that we're estimating in a GWAS, for example, are going to be biased by gene-environment correlation and other things like that. They'll tend, for example, which means that if we wanted to look at the SNP heritability and we've not included these controls in our GWAS, what we're going to get is instead of just looking at the fraction of the phenotype that's described by this portion, you're going to get the fraction that's described by this portion and some of what's here too. And so if you don't account for population stratification or assortative mating or indirect effects from parents or things like that, that's going to tend to increase your estimate of SNP heritability. And so that's another thing to keep in mind is, when we're getting our estimates of SNP heritability, what did they do with controls? Because that will affect how we should interpret the estimates. So, for example, here's some examples from the RDR paper by Alex that I was telling you about. So he used some data from Iceland. He estimated the SNP heritabilities at just a population level, not accounting for parental genotypes. And so these are so we call these like population level estimates of SNP heritability. And so for height, he estimated 55%, BMI 36, educational attainment, he gets 30%. He has age at first birth in women and age at first birth in men, so it's 20% and 12%. And so these are the estimates that are going to be biased by gene-environment correlation. And so they're going to capture sort of a little bit of this part here. Right? If you instead use the RDR method, which Alex developed, which accounts for indirect effects from parents and stratification and things like that, this is what happens to the estimates of SNP heritability by that method. And you can see that in general, these are smaller and sometimes much smaller. So height goes from 55 to 45. It's a little bit of a decrease, probably due to assortative mating. BMI goes down by a little bit. EA goes down by a lot. You know, the SNP heritability that accounts for parental genotypes is, it's cut almost in half. Age at first birth also falls by a lot in females, but in men by not very much. So again, this you need to be very aware of what type of heritability is being estimated. Yeah.
So am I correct in understanding that if you don't control for age and cohort effects, it won't bias the estimates? It will just reduce the heritability. It won't bias the data because it's just going to the error.
Yeah. So the question is, if you don't control for things like age and sex, then
Not even sex, age and cohort.
oh, age and cohort. Okay. Then is it the case that that's not going to bias the estimates? It's just going to make them more precise? And yeah, in general, that's true, right? Because you'd hope that age and birth cohort are uncorrelated with a person's genotype. If they are, that's a signal that there's some funny stuff going on in your data. And so if they're uncorrelated with the genotype, then they're not going to cause any omitted variable bias. But including it will absorb some of the residual variation and will make your estimates more precise. Yeah. So the things that you should control for that are important are things that are correlated with the genotypes, and that's why if you don't control for parental genotypes are correlated with your own genotypes, and so that can lead to bias if you leave them out.
So why isn't there more effort at like fine-tuning how you're modeling age for different traits and also adjusting for cohorts? Since it won't bias estimates, but it might improve our estimates of heritability, especially for something like age at first birth, for example, because of clear cohort trend.
So the question is, why isn't there greater efforts to do things like model age more carefully in things like GWAS? I mean, I don't know it's the case that there aren't efforts to do that when it's really important for the phenotype. It's a trade-off, right? Like if you could do more careful modeling, it generally requires additional data. I mean, if you have the data, you may as well use it, right? There's a paper that came out recently that said, when we're controlling for like age, we should probably be controlling for more flexible functions of age than we are, because we're losing a lot of power if we just control for it linearly. So I think that people are aware of these kinds of ideas, and depending on what data you have, you should do whatever you can. Great. Okay, so this is just an empirical point. In general, estimates of SNP heritability tend to be about 1/2 as large as estimates of narrow sense heritability. I've kind of beat this over the head a lot. Why is that? Why is SNP heritability smaller than narrow sense? Fewer SNPs, right? And it says it right here. HSNP doesn't include the effect of all SNPs, and so it misses the effects of maybe rare variants or other types of genetic variants, like inversions or whatever kinds of things that we don't include generally in GWAS. But it's also possible that these estimates of narrow sense heritability are overestimated. We often get these from twin studies, and if there's non-additive effects like dominance or things like that, or maybe assortative mating, so it may be overestimated because of the way that we model in these kinship studies. On the other hand, it may be that these SNP heritability estimates are biased upwards due to things like gene-environment correlation. So I think that the relative size of these things is not totally clear, just because it's really hard. There's limitations to all the different types of estimates that we have. Yep.
It's fascinating to me that they're able to recover such heritability with height. And I'm wondering if any of that is, or that sort of case is applicable to any sort of social science phenotype that we might be studying. Because I'm thinking that height might be especially additive and not have epistatic effects or, you know, more societally constrained associations. Do you have any intuition on whether there's an upper bound for the amount of narrow, or like, if there were, even if we can get all the additive variation out of narrow heritability for a behavioral trait, if it'll ever really match up with the SNP heritability?
Yeah, so the question is, you know, it's interesting that for height, we seem to be being able to bring together the narrow sense heritability and the SNP heritability. And, you know, is there any chance that we'll be able to see the similar sorts of patterns for behavioral traits? I'm not totally sure. The evidence that we have are things like, you know, we've done this dominance GWAS, and it seems like dominance effects are not important. And so, you know, the distance between, you know, so some people argue that distance is due to things like dominance, and it looks like at least dominance among common variants doesn't seem to be playing a big role. But I don't know, you know, what makes up the rest of the gap is kind of a mystery. I don't know if you have thoughts, David, about beyond that. Yeah, it's mystery to David too.
You mean that specifically within the context of EA?
Yeah, that for EA as well. Yeah, I mean, it's an empirical question. I don't know that there's you know, a lot of theory as to what we might expect in terms of how much would be additive versus non-additive and things like that. I mean, there is some theoretical evidence that tells us that we should expect things that are important to be additive. I don't really know those are, but you know, Peter Fisher always tells us, yeah, it's if things are have ever been selected for, then an additive model tends to be what arises naturally. I don't quite understand those points very much, but there are kind of quantitative genetics, population genetic kind of reasons for us to expect things to be mostly additive. Yep.
I was wondering, like, how, like, potentially, like, mapping the entire epigenome for a wide range of traits would allow one to understand, like, impact of, like, the environment and potential interaction with the genome, and maybe that might contribute something towards, like, narrowing the gap.
So the question is if there's a way that mapping the epigenome would help us maybe better understand these genetic effects?
I think to an extent that, like, you have, like, interaction between gene and the environment.
Mm-hmm. Oh, and again, specifically if there are interactions between the genes and environment. You know, I am not an epigenetics expert. I do know that, you know, people who do that kind of research, you know, do have high hopes for thinking about how the epigenome might tell us something about how gene-environment interactions may operate. But I'm going to punt to Lauren Schmidt, who's going to talk to us about epigenetics next week. I mean, it seems like it would, because, you know, we know that the epigenome affects how, you know, how genes are expressed, and we also know that the epigenome is affected by environmental factors. And so, you know, it's not crazy to think that you could use that information to start thinking about the kind of biological processes that link genes to behaviors.
Just wanted to follow up on Ted's question, because I think it's such an interesting question, but also like for something like educational attainment, we know that rich families are going to make up for the gap. You know, so what we know from educational literature is that, like, even kids from rich families with low test scores do quite well on their actual education attainment. So you will always have that gap, right? Like between how much genes can predict. Because kids from poorer families, even with good test scores, don't always go on to complete college because of environmental factors.
So that would be, okay, so the question is, you know, if you think about educational attainment, you know, wealthy families are able to fill the gap. So if there's someone who's underperforming, they can provide resources that kind of raise them up. And if the reason they're underperforming is potentially due to genetic factors, then you know, maybe that's you know, like that might be a model by which genetic factors might matter less in high SES families. So I'm not. So what I'm talking, the gap here is the gap between how much genotyped SNPs explain versus all SNPs, or all SNPs versus all combinations of SNPs. And what you're talking about, I think, would be more reflective of like how big is heritability in the first place.
Yeah, I was thinking in terms of like with height, we may be able to explain it really well eventually with good models or better data. But even if we were to sequence the full genome, we may never be able to. Yeah, I mean, I guess like the heritabilities. Yeah.
Yeah, I mean, I think that the issue is that our intuition maybe that understanding what the SNPs are doing for height might be easier because we think of that as more proximal. To you go gene to height in perhaps very few steps, whereas for education it's very, very complicated. How the SNP is related to the outcome in the end and can interact with the environment in complicated ways. But again, this goes back to interpreting SNP effects and what the magnitude of heritability is more than the gaps between additive, like SNP heritability and narrow-sense heritability. Yeah.
Question from John. Could dominance be more a factor for the SNPs that aren't measured or for rare variants? And are there any studies or suggestive evidence about it?
Yeah, that's a great question, actually. So you could imagine that like if there are really rare variants with large dominance effects, like we wouldn't see them very frequently in the population because maybe they get selected out if it's like a really, really bad thing. And so it doesn't contribute much to the variance, but maybe it's very important. There's a little bit of work on so where you might actually see these dominance effects show up is in inbred populations, because you see big long runs of homozygosity and things that might just go away because of selection in the normal population. It's possible to get 2 copies of the allele that's risk increasing, and I think that so there is some empirical evidence that among rare variants, perhaps dominance plays a role based on looking at inbred individuals. I'm not a really big expert on this, but the ultimate answer is yes, but I couldn't go much into the details beyond that. Alex might have something to say beyond that, or maybe not.
Not really.
Okay, great.
To follow up the Tomcat question, I don't know whether it's possible, but if we divide the population into wealthier families and the poor families, then take a estimate the SNP heritability, then would they differ quite a lot or pretty much similar?
Yeah, so one of the papers that I'm going to talk about later actually does some version of that, and they point out that actually it looks like the genetic architecture of education in high SES versus low SES families, might be different. There's some other technical issues with that particular model, but yeah, I mean, these are G by E questions. We're going to talk about G by E next week. I mean, it does seem, given that genes operate through the environment, then I think it's unsurprising that the effect of some SNP in one environment will be different than in another. So there's been some work on that. I think it's a very interesting question and kind of at the root of what a lot of us think about as social scientists. Dan.
There was a question before about the variance explained by dominance effects of rare SNPs.
In rare SNPs, yeah.
It seems like the kind of statistical argument for why dominance effects in general should explain little variance would have especially strong bite in the case of rare variants. So that you'd think, based on that kind of argument, that there shouldn't be much.
Yeah. So in terms of total variance, that's a good point. So in terms of total variance explained by rare variants, a rare variant by definition can't explain much of the variance because it's very rare. But just because it doesn't explain much of the variation doesn't mean that it doesn't have big effects. So there's this distinction.
But especially for dominance effects, because of the point that you're just going to have almost no data in the homozygous rare group.
Yeah. So if a SNP is only observed at all in 0.1% of the population, then you're only going to observe a homozygous individual with that SNP in 10 to the minus 6, I guess, percent of the fraction of the population. So it's you just don't see it very often.
What about epistasis? When does rare variants interact with each other more?
Well, there's an analogous kind of argument in terms of variance explained. That if it's rare, then you wouldn't expect. It's extremely unlikely for the same person to have both of the rare alleles. So even if it has a big effect, it's not going to explain much variance.
In the case of epistasis, though, there's you know, n squared as many interactions, though, right? So you know, any individual interaction would likely be unobserved really infrequently, but there's going to be a lot more of them. So that's a thing too. So I think that, you know, there's potentially more room for interactions, like technically, but I don't know of any empirical evidence because it's just so hard. We just don't have enough data to really dig into this very well.
Well, for height, the estimates are that I think epistasis does contribute with some of the variance, whereas the dominance doesn't contribute any.
Mm. Great. Okay, let's keep going. Okay, so we've talked all about SNP heritability. Now let's talk about polygenic indexes. And so polygenic indexes have become, I think, a lot more prevalent in social science research. This is a figure that was put together by one of our research assistants, Nancy. And what she did is she looked up every year of the BGA meeting, and she just poured through the abstracts and tried to identify the fraction of papers that were based on, you know, twin family and adoption studies in green, that were based on candidate gene studies in blue, and which seem to be using polygenic indexes in red. And so you can see in the last, you know, 10 years or so, the you know, prevalence of papers that use polygenic indexes has been, you know, rising steadily. Candidate gene studies has been decreasing. Looks like family-based studies is going down slightly. I wonder if this is going to go up again now that we've sort of realized how important gene-environment correlation is. But yeah, so these are becoming increasingly important, and they're, you know, really valuable and relatively easy tools to use. Not necessarily easy to interpret, but they're widely available. Okay, so let's start with the additive model. So, you know, we told you that the phenotype Y is, you know, there's some fraction of the phenotype that's explained by genotype SNPs, and then everything else that's left. And this is just another way to express the additive SNP factor. And so I'm going to refer to something as the true polygenic index. And this is like, you know, if the data gods were to descend and tell you, you know, exactly what these little bees were, you know, and they said, these are the bees that you should use so that you can maximize the predictive power of your polygenic index. You know, it would give you the same bees as these bees up here from our model. Then this is the true polygenic index. It's the optimal polygenic index, right? Any other bees that we use is going to be worse than this, just by the way that we defined what this additive SNP factor is. Okay, so here's sort of, you know, what we're aiming for when we're creating a polygenic index. So the variance explained by this true polygenic index is the SNP heritability. Why is that unsurprising? Yeah. So I mean, like we've defined the true polygenic index as the same thing as the additive SNP factor, and the additive SNP factor explains the SNP heritability of the variant guide. So these are just very, very closely tied concepts, right? So then, what is a polygenic index? Well, we don't have the data gods descending on us, and so we just need to do as best we can. And so what a polygenic index is well, let's estimate what these bees are based on the data that we have available, and let's just do a linear combination of the SNPs with our best guess for the bees. And so this is what the polygenic index is. So this is a thing that we can observe. This is a theoretical thing that we would love to observe if we could. Grant.
The little bees without the hat should not be interpreted as causal effects necessarily.
Yeah. So these aren't causal effects. These go back to the picture I showed you here. Sometimes I get excited about going back. So these are the these are the causal effects, but the little bees are these effects, you know, because there's omitted SNPs. And then these are estimates of those little bees. Great. So one thing to think about. So we're using our best guess, and what that means is that these little b's are if we've used hopefully we use some method to estimate the little b's, that we can think of them as like the true b, the b we want, plus some error. And hopefully we can think of this error as something that's mean 0 and is maybe uncorrelated. This should probably be a little b, is uncorrelated with the ideal weight that we'd want to assign the SNP. Right? So I'm gonna assume that we have some procedure that produces b hats where the error has this property here. Well, in that case, the polygenic index is just equal to this sum. I'm going to replace beta hat with true b plus error. And then if I distribute this x through, we can actually break this up into two pieces. So the polygenic index is equal to the true polygenic index plus some error. And because each of these u's are mean 0, then this error here is also mean 0. What that means is that our polygenic indexes you can think of as an estimate of the truth, and the expected value of our polygenic index is equal to the truth. There's a little bit of, once you rescale, we often first rescale our polygenic indexes to be variance 1, and that messes with this property here. But just for simplicity, let's keep operating under this assumption. Great. Okay. So we have our polygenic index. How good is it? So we refer to kind of the performance of our polygenic indexes by talking about their predictive power. So imagine that we have our phenotype Y, and we have our genotype data. We create a polygenic index, and we regress Y onto the polygenic index. Well, this is a simple regression that I'm describing. And so, the OLS coefficient associated with the polygenic index is going to be the covariance between the polygenic index and the phenotype divided by the variance of the polygenic index. All right. I'm going to now substitute in, the polygenic index is equal to the true effect plus error, and the phenotype is equal to the, or I guess I should say the additive SNP factor plus error, and the phenotype is the additive SNP factor plus the residual. Okay. And in the denominator, I'm just replacing a hat with a plus u, same thing. Well, u should be uncorrelated with a SNP and epsilon's going to be uncorrelated with u and a SNP. And so what we're left in the top is the variance of a SNP. And then because u is uncorrelated with a SNP, then we can just write the sum of the variance as the variance of the sum. And so this is the coefficient. If we were to do this regression, I'm going to kind of skip through the math here because you can just look at that on yourself. But then if we thought about what's going to be the R squared in this regression that I just described, you do a bunch of math, and you end up with something that looks like this. And so in the numerator, we have the SNP heritability squared. And in the denominator, we have the SNP heritability plus this term ME over N. So ME is the effective number of SNPs that we observe, the effective number of SNPs that we use in our polygenic index. N is our sample size. And I just want to talk a little bit about the behavior of this first. So imagine that this is the sample size of the GWAS that we use to create the polygenic index. And so imagine that our GWAS was enormous. It was like the population of the universe or something. Population of a homogeneous, randomly mating universe. And so n gets very, very, very big. And so this term here is going to go to 0. And so in the numerator, we have h SNP squared. In the denominator, we have h SNP. So one of those cancels. And what we're left with is just the SNP heritability. And so this was the point that I was making before. Or did I make this point yet? I don't know if I did. So the predictive power of the polygenic index is going to be bounded above by the SNP heritability. And as you increase the sample size, it's going to approach the SNP heritability. On the other hand, imagine that we did a GWAS in a really, really small sample, like one person or something. ME is generally assumed to be something on the order of 60,000 SNPs. And so this term now is going to be very, very big relative to these heritability estimates, which were these are bounded above by 1. And so what that means is if our GWAS is very small, then the predictive power of our polygenic index is going to also be very, very small. And so, so as you're creating your polygenic indexes, you want to be using large, well-powered GWASs to create these predictors.
So in year 4, you guys identified more SNPs. I'm assuming that the sample size also increased, and so the factor is the factor NE over N is not increasing. So it's not like you went from 1,200 SNPs to
That's actually a really good question. So this goes back a little bit to, so imagine that we did a GWAS in a very few SNPs, right? A very small number of SNPs. What's going to happen to the SNP heritability if I, really reduce the number of SNPs that, that I'm using to calculate SNP heritability? It's going to be smaller, right? Because the fewer SNPs we have, the lower the SNP heritability. So this will go down. On the other hand, the effective number of SNPs is also going to go down. Because if we only use 5 SNPs, then ME will be equal to 5. And so, there is sort of a relationship as we reduce the number of SNPs. It's not totally clear that the predictive power of the polygenic score is going to go down. This goes back to actually Rågr's question too yesterday, where he was saying, well, what if we just restricted to SNPs that we believe are going to be associated in the first place? Well, on one hand, the SNP heritability will go down if we just look at certain types of SNPs. On the other hand, this ME is going to go down, which will cause this whole thing to go up. So there's trade-offs. Yeah.
So how is the relative number of ME and N? So we know in terms of SNPs we haven't typed or genotyped everything.
Yep.
So we have limitations on that. But how is the relation? So when you increase your sample size, how is the effective SNP change? I mean that relative that ratio would dictate the predictive power.
Yeah. So if you increase your sample, so the effective number of SNPs is a function of the SNPs that you use and the population that you draw your data from. So if you could increase your sample size but not have to so this is why when I said a bazillion people, we're limited to, so if we were just drawing people with European ancestries, for example, or classified as European ancestries based on their relationship to the 1,000 genome populations, right? Even if we continue drawing people, ME should stay the same, because that's just a function of the SNPs that we drew and the LD between them, right? So that won't matter. If all of a sudden, though, we increased our sample size such that now we have people with different ancestries, that may have an effect on something like ME. But yeah, so these they're not totally independent, but theoretically we can think of them independently.
So ME is sort of mostly stable, unless you specifically intentionally choose to look at specific.
Yeah, if you hold your SNPs constant and your population constant, ME should be constant. Those are the two things that affect ME is the SNPs that you're using and the LD between them. So when I say hold your population constant, I mean draw from people who have the same LD structure. Good? Yep.
Sorry, just a quick question. What exactly do you mean by predictive power here? To me, R-squared is just a measure of fit. So.
Yeah. So by predictive power, I mean R-squared. So so R-squared can be interpreted as the fraction of the variance in the outcome that's explained by the variables in the model. And so that's a measure of fit in the sense that if your covariates explain all of the variation in the phenotype, then your model fits very well. So that's kind of the relationship between the two. Does that answer your question? Okay.
Muin is asking to elaborate how you get NE over N from the previous line, so from variance of UIs over variance of Ys.
I could do that. I'm just thinking, but it's not a super complicated derivation, but it would take 5-10 minutes, and I think that it probably, I mean, unless people are really excited. Are you really excited? I'm not sure that, yeah, I can talk to Moeen about it after if that's okay. I mean, the idea that where it kind of comes from is that this piece here is just related to how much error there is in your betas. Like what's the sampling variance of the little b's? And sampling variance is decreasing in n. And so that's why the over n shows up. And the amount of error is aggregated over all the SNPs that you use in your data. And so if you use a lot of SNPs to create your polygenic index, then you're adding error on top of error. And so that's where this ME comes from. The more SNPs you add, the more the error can compound across the SNPs that you're using in your PGI. That's like the intuitively where it comes from. But we could also just work through the math at some point if he wants. Yep.
So I have an extra question about how you estimate the beta hats.
Yeah.
In that regression, are there any controls?
Can I punt on that?
Sure.
Because yeah, we're gonna talk concretely about where the B's come from in a few slides. Yep.
Just for clarification, the polygenic index, the SNPs included to the estimation of polygenic index ranges from 1 to k.
Mm-hmm.
So that basically means all SNPs we observe in the G-verse estimates. Like yesterday, we also saw the kind of Manhattan plot. Then some SNPs reaches the threshold, when
Mm-hmm.
other SNPs didn't reach.
Yeah.
But my understanding is polygenic scores includes everything,
Yep.
regardless of the genome-wide statistical significance.
Yep.
Is that right?
Yeah. Is there any more questions? We're gonna talk in detail about the concrete how we select the SNPs that we include, how we pick the weights. We're gonna talk about that in just a few slides, I think.
Right.
Is that okay? Great. So here's just a little picture that I made once that shows this relationship between the sample size of the GWAS and the predictive power of the polygenic index. And so if you see from this formula, you have ME, which is like a fixed constant in for a particular population. And so after that, it's just a function of the SNP heritability and the sample size. And so this is what we're showing here. So imagine that we have a trait like height, which has a SNP heritability of about 50%. Well, the expected R squared, based on our Detweiler formula, if we had, so this is sample size times 10 to the minus 6. So 1 million people, we'd expect the R squared of the height PGI to be like 40% for height. And eventually we'll asymptote up to 50%. You can kind of see if we went further this way. You can see it's starting to approach that line. For something like EA or BMI, the SNP heritability is closer to maybe 20%. And so in a sample of 1 million people, maybe we'd expect to see around a 15% R squared or something like that, and increasing up to the 20%. So when we're at 2.5 million, maybe we're at 18 or for something like subjective well-being, the SNP heritability is closer to 8%. And so this is rising up much more slowly. So even at a sample size of 1 million people for subjective well-being, we're only getting about half of the SNP heritability there. This figure was made, I think, 4 years ago. And we're actually getting to the point that we're actually to the right. Imagine someday we have 2.5 million people. The GWAS of educational attainment that's going to be coming out soon is 3 million people. So we're right here. And I think the GWAS of height that's coming out is even more than that. So I'm going to have to update this picture.
Is the idea here that we're kind of approaching the true heritability?
At least for things that are quite heritable. I mean, you can see that the rate that this increases is also a function of the SNP heritability. And so we're at 25%, and we're at half of the SNP heritability for height, in samples of a few 100,000 people. We are only at half of the SNP heritability for subjective well-being, at 2 million. And so the rate that this increases is also going to be a function of how heritable the phenotype is. Yep.
I understand for some of our EA GWASs, which I haven't really been a part of, we M-tag them with other phenotypes.
Yeah.
Do we make scores off of, or indices off of those M-tag results?
You can. So Alice was asking about what if you, so there's a method called MTAG, which allows you to meta-analyze summary statistics together for GWASs of traits that are not the same thing, but are maybe genetically correlated. And in general, you see that boosts predictive power. It boosts effective sample size, because your standard errors are tighter, and so your error might be smaller. And you're saying, can you make polygenic indexes out of those? And yeah, we do. And on average, we do see increases in predictive power for those polygenic indexes. There's some nuance as well if you're doing something like that. We discuss it a little bit in the PGI repository paper. We have it's especially specifically in the user guide. But the issue is that there's some contamination that can happen across phenotypes if you use MTAG and create a polygenic index out of those summary statistics that make it a bit more tricky to interpret results.
Right. What I wanted to ask is, like, does that, if we were to hit this theoretical limit with MTAG results, are we still hitting it for purely EA? And it seems like maybe not.
Yeah, and that's the issue, right? Interpreting the results if you're using something like MTAG is a bit funny.
Okay.
So I think Rågr and then over here.
Yeah, I was wondering, like, if we could maybe draw like same chart, keeping n constant, but increasing the effective number of SNPs. Basically that has happened in terms of like the over time effective SNPs have increased, right?
Yep.
How does it look in terms of the predictive power?
Well, so in principle, it's going to look like this, just looking at this equation here. But I don't know. I also don't know, like including rare variants would increase ME a little bit, I suppose. I don't have a good sense of how ME has changed based on the set of SNPs that, it used to be that we used only quite common SNPs, like, greater than 5%. And now we go down to like 0.1%. So maybe that ME is not actually very good. Like you don't use the same ME for the summary statistics that we used before as we do now, but I'm not totally sure. I mean, you could do it, and it would look like that, but I don't know what a reasonable range is the problem. Yeah.
So we recovered the true SNP heritability for height. Is that true for just European ancestral groups, or like other different admixed or the different ancestral groups?
Yeah. So the question is, if we've recovered the true heritability, SNP heritability for height, is that true for other ancestor groups? And the answer is no. Pretty much all of the, a huge fraction of the genomics work that has been done has been just in individuals of European ancestries. And because the predictive power of the polygenic index is a function of sample size, the sample sizes in other populations is much lower, and so we're much further in this direction. There are methods under development right now, things that have recently, like PRS-CSX that came out, where they say, well, what can we learn from GWASs of individuals in European ancestries to increase the predictive power in other ancestors? This is a super active area of research, a super important area of research too. Great. Should we keep going? Okay, so we've operated here under this fantastical idea that there's these B's that are real and they're fixed and they are the same for everybody, right? In fact, that's not the case. I mean, some of you have even asked questions related to this. So it may be that if we look at a group in the UK, that the little b's are different for the UK group as it is for someone from Australia, and different for someone from Arkansas, or or someone who was born 40 years ago. Like, there are lots of reasons why we might think that because these little b's operate through the environment, maybe they change from cohort to cohort that you're trying to predict into. And in fact, the data that someone cited earlier is if you look at the genetic correlation of educational attainment between the UK Biobank and 23andMe, it's like 0.7 or something like that. So it's definitely the case that there seems to be some heterogeneity in the genetics of educational attainment, at least, between different groups. So how does that then affect this point was also brought up by this paper, Mostafavi et al., where they look at predicting educational attainment in high SES individuals and low SES individuals, and they show that the predictive accuracy of the polygenic index is different between the two groups. So how does that affect the math that we did? So let's assume that we have some other cohort, and we for maybe a different birth cohort, for example, and so a star SNP is going to be the additive SNP factor for our phenotype in the alternate cohort. And so Y star is the phenotype in the alternate cohort. So because we've assumed there's maybe heterogeneity, that means that the additive SNP factor in the star cohort might not be the same as the additive SNP factor in the initial cohort that we've been working with. And then what that implies is that the SNP heritability in the star cohort may also be different than the SNP heritability in the other cohort. And furthermore, the correlation between these two things might not be perfect. It might be something less than 1. So under these assumptions, what happens if you create a polygenic index in a sample, in non-star sample? I feel like this Heritabilities story or something. But in sample 1, and then we try to predict our phenotype in the starred sample. Then what's going to be the expected predictive power there? Well, you do a bit of math, and you get something that looks a bit like the Detweiler formula. We have things about heritability in the top, also in the bottom. We have ME over N over here again. Right? But it's a little different. So there's a few things that are important. So notice, for example, this RG squared exists up at the top. And so if the samples are very heterogeneous, then RG is going to be small. And so that would decrease the predictive power. And so to the degree that there's heterogeneity between the two samples, the polygenic index is not going to do as well in the other sample. However, we see that the heritability in the target sample shows up here. And so let's assume that the heritability in the starred sample is bigger. For some reason, just the phenotype is much more heritable in sample star. What's going to happen to the predictive power in that case? So I'm saying this is bigger, so this is going up. Yeah, so it'll actually increase. So there's actually a chance that even if there's heterogeneity, if the increase in the heritability is larger than the decrease due to the heterogeneity, like it's possible that you actually could see even more predictive power in the starred cohort than the other one. And so it's not always predictable which direction it's going to go, even if you think there's heterogeneity in effect sizes. Kind of one final point is, this would be true. Like, let's say that Y was not even the same phenotype in a different cohort, but it was just a different phenotype. We saw this, for example, if you take the polygenic index for educational attainment and you predict cognitive performance. Cognitive performance is a much more heritable phenotype. So even though there's heterogeneity, there's the genetic correlation between cognitive performance and education is less than 1. The increase in the heritability swamps the decrease due to heterogeneity, and you actually you get greater. The educational attainment polygenic index predicts cognitive performance better than it predicts educational attainment. I assume that's still true. That was true for like EA2. I haven't checked recently. Great. We happy? What time is it? 12. We're good. I think I have 10 minutes before lunch is scheduled, right? Cool. Great. So I'm going to quickly go through a handful of other points before we break. So one of the major advantages of polygenic indexes, so we talked initially about candidate gene studies. And we said one of the big problems with candidate gene studies was that they had such small effects. They explained so little of the variation. It meant that studies tended to be underpowered, and led to all of the problems that Dan told us about. Well, PGIs can actually be quite well powered, and so because we're aggregating the effect of a bunch of SNPs all together, then a PGI will generally have much more predictive power than just a single genetic variant that you might see in a candidate gene study, for example. And so here's some examples of estimated R-squareds for PGIs. These are the same phenotypes that I showed you before. One quick distinction. These estimates came from the Icelandic sample. These came from either the HRS or AdHealth. So these aren't necessarily comparable. Also, all of these R-squared estimates are based on samples of unrelated individuals. And so to the degree that these two are comparable, this column is comparable to the population SNP heritability estimates. But you can see that in general, these are smaller, which we expect, right, because there's noise in the PGI. So for height, we're explaining about 24% of the variation. We're 24, which is almost half of the total variation, of the total SNP heritability. Sorry. BMI 36 is the SNP heritability, and current polygenic indexes can get us up to maybe 10%. Educational attainment, well, this 30% is in Iceland, so this is where things are a bit fuzzy. But for polygenic indexes in AdHealth, we're getting things as high as 16%. I think that the estimated SNP heritability in AdHealth is something like 40%. It's really big for some reason. It's a bit of an outlier in terms of heritability. But still, 16% is what we can get in some available samples. For these two fertility measures, I couldn't find the predictive power split by sex in this paper. But when they combine it in AdHealth, they're getting about 5% in terms of age at first birth. So that's getting to be pretty good. When I updated these slides for this lecture, and this was 1% when I presented this last time. So we're just seeing, as these sample sizes have been getting bigger, these polygenic indexes have been getting stronger and stronger. So this figure, which is too small for you guys to see, but if you squint and look at the slides later at home or something, what this is, it's a figure that was put in the paper that introduces an SSGAC project called the Polygenic Index Repository. What we've done is we've taken the biggest GWASs that we could find for a bunch of different phenotypes, including data from 23andMe, which is not easily available to researchers. And then we collaborated with a bunch of different data sets that provide genetic data to social science researchers. And we constructed polygenic indexes for all of these phenotypes using a uniform methodology. And so this is just reporting, I just mostly wanted to show you kind of the class of things. So we have, BMI and height. We have a handful of cognitive PGIs that are available, things related to fertility, a bunch of health behaviors, personality, and well-being. So there's a lot of phenotypes available. Through this collaboration, what we do is we provided these PGIs to the data providers. And so if you wanted, for example, to use these PGIs from the PGI repository in AdHealth, then you can apply for that data from AdHealth or HRS or whichever data set that you're using if they're involved in the repository effort that we're doing. And so hopefully this makes your life easier. You don't need to actually go in and try to make the polygenic indexes. It makes things maybe more comparable. So if you're using the polygenic index in HRS and AdHealth, those were constructed in the same way. So hopefully this is a valuable resource to you guys who are using these data.
The y-axis is incremental r-squared. What is it incremental over?
So it's just incremental over the set of covariates that we use. In general, that is age, sex, age squared, interactions, a handful of PCs. If the specific cohort recommended additional controls like batch effects or things like that, we would include those. But nothing crazy. It's not like we're saying incremental over SES or things like that. It's just simple controls.
I have a question about, I'm interested in looking at differences in monozygotic twins, and so I'm kind of conflicted about kind of conceptually and statistically incorporating polygenic risk scores, because in theory, because monozygotic twins are identical genetic-wise, it doesn't make sense to include polygenic risk scores or differences between the polygenic risk scores as a covariate. But considering about how we know how gene-environment correlations could bias SNPs, statistically, they could have differences in polygenic risk scores, right?
Okay, so the question is about, if you're using monozygotic twins, then they have the same genes for the most part, all the same SNPs, and so, it maybe would be hard to do research like polygenic index research in those groups of individuals. But like, if there's gene-environment interactions and the twins are facing different environments, then maybe they would have different polygenic indexes. In practice, when we construct polygenic indexes, we use the same weights for everyone. They're environment blind. And so I think you're still, based on current practices, you're still in trouble with that. I mean, I suppose if we got to the point someday that we had really fine-grained estimates of what the effects of SNPs are in different environments, like maybe something would be possible, but I feel like that's a ways out. I don't know that's going to be something that's possible to do very soon. Great. Here's just one more picture illustrating the Detweiler formula that I was showing you before, showing the relationship between sample size and incremental R-squared, predictive power of the PGI. And so these sets of dots are PGIs based on different iterations of the educational attainment GWAS. And so these two dots here is a PGI based on EA1, which was in a sample of about 100,000 people. These sets of 4 dots here are based on the EA2 discovery and EA2 combined discovery replication. These two are EA3, and this is going to be for the upcoming EA4. And we've predicted educational attainment in the AdHealth sample in the health and retirement study. And you can see that there is a steady increase, so that's great. I told you that as you increase sample size, predictive power should go up, and it does. The dashed lines here correspond to predicted, based on these theoretical formulas that we showed you, what do we predict the predictive power to be? And at least in HRS, it seems to follow pretty well. AdHealth's doing really well for some reason in our current data, and I don't think we understand why, but yeah, in any case, that's that.
Yeah, I was going to ask why get.
Yep, that's a great question. I wish I knew.
Is this because AdHealth is doing good, or HRS doing worry for the newest?
Well, I don't know. I mean, what's puzzling, right, is that, these are all AdHealth, right? So if you think it was something about AdHealth, then all of these SNPs, all of these points would be higher, right? But there's something about AdHealth with the EA4 data that just makes it it's puzzling. I don't know. Dan.
AdHealth is relatively younger cohort.
Sure. But that would affect that point too. It was a young cohort for this point as well.
Maybe more of them are using 23andMe. Yeah.
So possibly it's overlap. Is that what you were going to say? Yeah. So if AdHealth is all signed up for 23andMe because they think that sounds fun, then that could bias upward these R-squareds.
Actually, even just a small fraction.
Okay. Yeah, that's true. Even a small amount of overlap can cause big problems. So maybe that's what's going on. So maybe we're a little optimistic here just because we can't distinguish the overlap between these samples.
But didn't you use LD score regression at some point to look at this?
I mean, you could try. So Raymond showed us that if you use the LD score regression, you can get estimates of sample overlap between samples. The problem is that AdHealth is not very big, and so we're not really well powered to estimate the overlap.
Could it be because in the HRS cohort, the older women, their actual educational attainment is lower than the people with
Yeah,
higher score?
I mean, that might be what describes, why systematically HRS is lower, and I think the SNP heritability in HRS is lower, and so stories like that might explain the differences in heritability between the cohorts, but I don't know what explains that gap unless it's a sample overlap. That's honestly probably what it is, because I can't think of what else it might be, but maybe.
Do these cohorts keep track of participants that have signed up for 23andMe?
Oh, that'd be cute. I don't think so.
I mean, they ask for a lot of information. It'd be interesting to see if they.
Yeah, that'd be useful. I mean, the issue is they would need to know if they'd need to know if they signed up for 23andMe, and also if they answered the survey question that was used in the results that 23andMe gave us.
Patrick, maybe you should say something about why sample overlap would create inflation of the R-square.
Yeah. Yeah. So so the problem is the little bees that we use, the Let's see, go back to a slide that shows these little b's. Oh, this one doesn't even have it. So these b hats that we use, we decompose it into like little b and the and the error. And when we were going through the Detweiler formula, I just asserted that the error in our estimates was independent of the phenotype residual, right? If it's the case that there's sample overlap, what happens is that U is going to end up being correlated with E, because the source of the error in our estimates is the residual variation here. And so that induces correlation, and so there's an additional term in the numerator, and that inflates the R-squareds. And so that's where it comes from. I mean, it's overfitting is also just sort of the shorthand term that you'll hear. So if there's sample overlap between the discovery and prediction sample, then you have overfitting.
Can you also tell us how you adjust for related individuals in that?
To get this number?
Yeah. Is it a similar kind of form?
So that would... So in general, I think of related individuals as causing problems because of the standard errors. So related individuals make your standard errors too tight if you don't account for them. But I don't think it creates bias in terms of the associations. And R-squared is just a function of the association and not the standard errors. So like, if I tried to draw error bars around this, then related individuals would be important. But we didn't draw standard errors around this because it was hard. Great. Okay, so I'm going to do this slide, and then we'll break for lunch. So this is just the same point here. PGIs are great because they're predictive, and so what that means is that we can get these estimates of little b hats in very large GWASs, right? And then construct PGIs in small samples. And because these PGIs are quite predictive, then we're going to be well powered to detect the effect of the PGI even in a small sample. So I have some calculations here. So if the R-squared of the PGI is 13%, which is what we see in the health and retirement study for the education GWAS, then we'll have 80% power to detect the effect of the PGI in a sample of just 61 people. So it's getting to be pretty small data sets, to be honest. If you think about the estimate of, this should be 16%, like 0.16 and not 0.016. But if the predictive power is 16%, which is what we see in ad health, then we only need 49 individuals to have 80% power. So this is getting to the point that we can now actually start using this in pretty small subsamples and start detecting the effect there using current PGIs. So that's really exciting, I think, and it really opens the avenues for the type of things that we can look at, because now we can look in data sets that have super high quality measures of outcomes, measures of mediators. We can include different covariates, and so suddenly we can answer a lot of really interesting questions now that we have some well-powered tool to study them. Okay, I told you all we're going to talk about constructing them. We're doing it after lunch.